Alright, let's continue on with Chapter 9. One of the first theories of color vision, of how it is that we perceive color, was proposed in the 1800s by Young and Helmholtz. And it's a relatively simplistic theory. So what they hypothesized was that there are three different receptors that are responsible for color vision. So there's a receptor for one wavelength of a light, a receptor for another wavelength of light, and a receptor for another wavelength of light. And the first bit of evidence that came about for this theory was behavioral evidence, where they did color matching experiments. And basically, what they did, what the folks did in this study, was they looked at a spot of light that was shining a certain color, and then their task was to adjust the relative brightness of three separate lights that are three different colors to match the color of the sample they were looking at. So they had these they would turn these dials and they would dim or brighten a light, and until the color matched what they were perceiving. So that was really the first behavioral evidence that we could use these three colors, that we could use three basic colors to essentially create any color that we can perceive. So from doing this study, it was discovered that people can do this, right? And you need at least three wavelengths, a short, medium, and long wavelength, basically. You need three wavelengths to be able to create a color. You can't do it with two, and four will work as well, but the minimum that you need is three. People that are colorblind or have other types of color deficiencies will perceive a different color from the sample, and then they will match that color using only two of the lights. They don't need three; they can do it with two, which suggested that people with that are colorblind or have other deficiencies in their color perception may be that way because they're missing one of the three receptors that they need. So here's an example, right, of the way the study was done. So the subjects would look at the test field in A, right, that's shown in side A, and then they would get the chance to adjust the brightness of three separate lamps that are shining at different frequencies. So the one, in this example here, they're looking at a light that shines at 500 nanometers. This should be an N, not an M. But they're looking at a light that shines at 500 nanometers. And then they have three lamps, one at 420, one at 560, and one at 640. And what their job is to adjust these lamps until they get a color similar to this. Normal people need at least three lamps to do this. Color deficient people that are colorblind, for example, can do it with two, but it also means that they're misperceiving this color. They're not seeing this color as normal sighted people are. Going deeper, there is some physiological evidence for this theory. In the 1960s, we discovered the visual pigments. Visual pigments are large proteins that live, that are found inside of cones, and these are the part of the receptor mechanism. So essentially, whenever a light photon traveling at a certain frequency hits one of these visual pigments, it begins a cascade chemical reaction that ultimately ends in you perceiving the color. But the folks that were studying these visual pigments starting in the 1960s discovered that within the cones there's visual pigments that respond the best to different wavelengths. As a matter of fact, they discovered that they were able to group visual pigments into three different groups. Ones that responded maximally to short wavelengths, another group that responded maximally to medium wavelengths, and then a third group that responded maximally best to long wavelengths. And later on in the 80s, it was the genetics of these proteins were found, such that they were able to link the creation of these proteins to specific genes. And the way the genetic layout worked was that they were able to discover that, or they discovered that at least in humans, that these genes were coding for, in general, three different types of pigments. So here we have a chart indicating the absorption spectra of three visual pigments. One that is absorbing in the short wavelengths, one absorbing in the medium wavelengths, and one absorbing in the longer wavelengths. So the ones that are absorbing in the shorter wavelengths here, 400-ish, about 425 nanometers, is perceiving blues. The one that's absorbing here in the medium wavelengths is about 525 nanometers, is perceiving greens, and then the reds and yellows are taken care of by this guy right here, which is perceiving the longer wavelengths, roughly about 575, and it responds pretty well also to the 600s and whatnot. So what this tells us that we can group these proteins, these visual pigments, into three separate categories, each of which responds the best to a particular wavelength of light, either a short wavelength, a medium wavelength, or a long wavelength. So what does this mean, basically? Well, it tells us that the response of the cones, I'm sorry, color perception is probably, or may be due, color perception may be due to the response of three different types of cones in our retina. So probably, or at least what this theory is talking about, is that these visual pigments, each group of visual pigments lives inside of a specific cone. So one cone, for example, is going to produce visual pigments that only respond to short wavelengths, for example. And all of these, so there's cones there's cones that respond to short wavelengths, ones that respond to medium wavelengths, and ones that respond to long wavelengths. Um, and these guys all work together to produce the perception of color. Um, and we've also have some evidence from color matching experiments that colors that are perceptually similar to each other, which we refer to as metamers, can be caused by different physical wavelengths. So you can actually have two colors that look very similar to each other, but they are in fact caused by different mixes of these three wavelengths. That's very possible, and is consistent with the with this view that color perception is based upon these three types of cones working together to produce color vision. So here's an example, right? This is a cartoon drawing of three of cones, right? So in this example here, we would have these three cones working together. This one responds maximally to short wavelengths. This one to to medium, and this one to very long wavelengths. Notice that among these three cones working together, The bigger one, the one that responds best to blue, or the one that responds best to the short wavelengths is this one. It's a stronger one. So what it's doing, um, what it's doing is firing stronger to blue. So this particular cone here fires very strong to blue. This one not so much, and this one very little. So that tells us this is a cone that probably detects light in the blue spectrum. Similarly, we have these three cones here. This one responds very powerfully to medium wavelengths, and this one kind of like a little bit of responding to short wavelengths, and this one a little bit of responding to long wavelengths. And these three combine together to create the perception of green. This guy right here, these three guys right here are working together to respond. This guy responds to very strongly to long wavelengths. This one not so strongly to medium, and this one hardly at all to short wavelengths. So when they all light up together, we perceive red. And when these guys all light up together, respond very strongly to long wavelengths, strongly to medium wavelengths, and not so much to short wavelengths, we perceive yellow. And of course, white, as you recall, white is all the wavelengths shining at the same time. So that's what we see here. We see the short, medium, and long receptors all being lit up at the same time. And when that happens, we perceive the color white. So here's another another example, right, of how this might work. So here we have two receptive fields that we're looking at. In this receptive field here, and again, going back, this is an example of how you can have the exact same color from different wavelengths. So in this example, we have two separate colors. Oh, I'm sorry, we have two separate receptive fields here. This receptive field is being hit with 530 nanometers and 620 nanometers shine in the same spot. This receptive field is being hit with 580 nanometers only. However, the perception is the same. And the way we know that is by looking at the outputs of these cones, right? When 530 and 620 hit these three cones, it causes the cone that responds to short wavelengths to fire very low, the cone that responds to medium wavelengths to fire in the middle, and then the cone that responds to long wavelengths fire a lot. And that's the exact same firing pattern that you see here. So the take-home message here is that the cones all work together, firing at different intensities based upon the wavelengths that are being hit, and they work together to ultimately create the perceptions of color based on how much they're firing relative to each other. So now we want to ask ourselves, do we need to have three receptor mechanisms? Do we need three receptor mechanisms for color vision? Well, yeah, pretty much the answer to that is yes. And the reason for that is because, well, you can't have color vision with one receptor. Because what's going to happen there is that, a receptor, a cone only fires, it either fires a lot or a little. And based on how many photons hit it at its correct wavelength, right? So the best you can do with one receptor, one cone, one color receptor, is to see lighter or darker versions of the same color, right? So if you have one receptor that is, if you have one cone that's tuned for reds, for long wavelengths, right? Reds. It's only going to light up when it gets hit by red light, and the brighter the red, the more that cone's going to fire, and that's all you're going to be able to see. So you're just going to see changes in intensity. You're not going to see changes in in color, you're just going to see changes in intensity. If I show you blue, you're not gonna be able to see that. It's just going to be black or white or whatever. There are people that actually there are dichromates that only have two receptors in their eyes. And with dichromates, they can perceive more colors. If you're a dichromate, you're going to have color deficiencies in seeing certain colors. Now you can see more colors, right? If you only have two types of receptor detectors, or cone receptors, I should say. But you see more colors with three receptor types.